part of the preliminary pilot study for the Microbiome project. So here we see samples from, again, I'm using very high-level environmental data levels, but we have samples from stream water collected in Colorado, samples from soil collected all over the US and various other locations, marine sediment from the coast of England, human skin and human guts and human mouth from the Human Microbiome project. The air, which is collected around Colorado again, coal bed, lake water, and other systems which provide us with information on the different ecosystems in which we're exploring. This is based on alpha diversity. So this is the kind of study. All of these projects were generated using the same DNA extraction method, using the same analytical approach to explore the data, and we can start to see that as these data are comparable, we can now compare these different environments in a more fundamental way. And Rob and I will talk a little bit more about this, but this is some chain analysis. So these are the 5,387 samples. There was an average of 39,000 sequences per sample. This is sequenced using the Illumina ITAG approach from Caprasso et al. 2010. And these are randomly sampled down to 2,000 sequences each. So we've said that every single data set has exactly the same number of sequences, the same number of observations per sample. So we have the same number of observations for 5,387 samples. And this is the three-dimensional space generated by a Bray-Curtis matrix of the community composition and the relative abundance of the community members in each sample. Out here we actually see some of the human microbiome samples from the gut, the mouth, and skin. Up here we have some of the marine samples and some of the soil samples down here. And then an awful lot of interaction in the middle. There was a recent study coming out of MetaHit, which obviously BGI has played a very significant role in generating, which suggested there were 3 enterotypes for microbial communities in human guts. So every single one of us may have a unique enterotype that describes our gut microbiota. Well, on the look of this, it would be very hard for me to describe a unique microbial enterotype for all of the environments that we've actually sequenced. Although we could put borders and gradients upon those. Now, I'm just going to talk a little bit in the last 15 minutes of my presentation about some studies that we've also been doing on different environments. This is an example of what we can do should we generate these kind of data. These are a suite of samples that were collected as part of the National Science Foundation of America's NEON study, which is basically a number of sites around the US that have been selected to represent specific soil-based biomes around the country. And they are now being regularly sampled as part of an ongoing long-term collaboration. So through some research by Dion Antonopoulos and Sarah O'Brien at Argonne National Laboratory, we selected a number of different sites representing forest environments, grassland environments, and we sequenced metagenomic data from these environments using the Illumina protocols to determine if there were significant differences in those ecosystems. Now, the results of this are pretty fundamental, albeit maybe not very exciting. But one thing you have to bear in mind is that nobody had shown this before. One thing that it showed was that grasslands are different to forests. They have a different microbiota, a different functional division, and I know what we do, that's terribly exciting, but this data set allows us now to interrogate why those differences exist. And one thing we found was that for the more than 800 different functional groups that we identified, most of them were related to carbohydrate metabolism. So we tracked this down further, and we see differential carbohydrate metabolism based on the root exudates, coming out of the grassland plants compared to the forest plants. So this has a massive implication when we deal with carbon transfer and flow in these different ecosystems. Once we understand the mechanisms by which carbon is flowing in grasslands versus forests versus tundra versus marine samples versus the human skin, we start to understand how that community functions and is deliberately different from the different ecosystems. Now, vegetation was very important in defining these systems. climate and geography had virtually no impact. So you could take two samples which were very close to each other, but when one came from soil from a grassland versus a forest, it didn't matter if they were very close to each other, they were very different. But two forest samples located very distantly were very similar. So that geography had no impact upon microbes' definition of where they lived. This is very important because it doesn't matter where you take the samples, the microbes exist along these environmental ecological gradients. And they live based in a physical, chemical world. This is some mammalian research from the Western English Channel. The English Channel is the busiest shipping lane in the world, or was, I think still is to a certain extent. For many years, the UK has been collecting samples off the coast of the southwest here, which represents a confluence of the North Atlantic and the English Channel. This represents a 6-year time series. This is 72 time points and a log of species richness over each of those 72 time points between 2003 and 2008. So every single month for 6 years. And what you see here is incredible regularity. The community is very robust in how it's represented at each time point. So what we decided to do was take one of those time points, and bear in mind we had 10,000 sequences per time point for 72 time points. We took one of those time points, December 2007, rather arbitrarily, and sequenced 20 million reads. And this is based on the Illumina ITAG protocol. And what we did is we found that 99.96% of all of the species identified in that 72 samples there were present in this one time point. This is pretty exciting to me, because what it suggested was that it doesn't matter which time point we looked at, the species present there were always present there. The species present in June are present in December. They're just different abundances. And the relative abundances of the community change, not the community composition. Now, this enables us, understanding these precepts, which you wouldn't be able to understand if it wasn't for Earth Microbiome Project style studies, enables us to start making mathematical predictions about those changes. And this represents a Bayesian network where up at the top here in these grey diamonds we have environmental parameters, temperature, salinity, chlorophyll concentration, etc. These environmental parameters can be used to predict the relative abundance of specific community members, the taxa in the environment. So each one of these cells down here represents a specific taxa and its change in relative abundance over observed time, represented by this formulation. Now, what we can use this to do is predict and extrapolate out microbial diversity for this ecosystem. This is based on a fundamental understanding that the community composition never changes. So up here we have 6 years worth of observed data. These data are the same. This is the actual recorded observed relative abundance of the taxa in the system. And here we have the prediction of that community. It's extrapolated out over 10 years. So where we have overlap between our observed and our predicted community, we can calculate how accurate our predictions are. And they are, on the whole, between 95 and 100% identical. So the community, the predictions of our methodology is highly validated. It's fantastic. But what it enables us to do is extrapolate beyond our observations. The design of many of the Earth Microbiome projects will help us to extrapolate beyond our observations, to predict into the future, and to hindcast into the past. The metabolome is also very important, and Rick showed this what we call the hairball. And the hairball is for a particular ecosystem, is all of the metabolic interactions and metabolites generated by that community for that given system. So every single one of the dots and the nodes in this plot are metabolites, and the lines are environmental, actual enzyme activities. So what we do is we predict the enzyme activities, and changes in relative abundance of those enzyme activities can be used to predict the metabolites and their concentration in the system. So what we see here is, for example, carbon dioxide, and this is over 24 hours. So a sample taken every 4 hours over 24 hours. And what we can use this system to do, this is metagenomics, mind you, this is a metagenomic profile of the community. And what we see here, another revelation in terms of microbial ecology, is that the community consumes CO2 during the night and generally produces CO2 during the day. So when it goes yellow, it's during the day, and when it goes dark, it's during the night. We call this term photosynthesis. We find it to be, we're hoping no one's published this before. We're trying this through science now. We haven't had any comments back. But I know it's obvious. It's very obvious, but you couldn't do this before. These are metagenomic data that can be used to predict the environment in which the microbes come from. That is fundamental. Because what it means you can do is you can take those environmental parameters throughout your ecosystem, and you can use that to predict the community composition of your ecosystem. That, via a linear relationship, can be used to predict the metabolites that can be generated by that community and consumed by that community. And these metabolites tell us what the environmental parameters were like in that community. It's a wonderfully cyclical synchronicity, and I love it. So essentially, what we're now doing is we're actually taking, this is a satellite image of my new home, Lake Ontario. No, where is this? Lake Michigan. See, Illinois, where I come from now, doesn't even have its own lake. This lake is named after another state. Go figure that one. So Lake Michigan, we can tell from many of the environmental parameters, we can tell by using satellite data, many of the environmental parameters on the land and in the water of Lake Michigan. And by generating sequencing data from the microbial populations in different locations over time series, we can use that satellite data to drive this prediction of microbial diversity. Again, we can use that to infer via metagenomic and metatranscriptomic studies the community profile of the metabolites, which then can be used to validate the predictions from the satellite data. So this is a very 1980s style cartoon of the changing microbial diversity in the English Channel. Now this little box up here in white is the site in which we actually went in and did that 6-year study of microbial diversity. So what we're showing here is based on satellite data for every grid square in this prediction, we have predicted what the microbial community composition would be relative to L4, so this one little site here. So when the community comes into winter, and we're starting to see that now, it's in autumn, we actually see the community is incredibly homogeneous. Black or dark blue means it's homogeneous, it doesn't really change. And we go into spring and summer and we start to see a lot of differential variability in the community over this spatial scale. Again, this is the southwest of England, this is the northern tip of France.